Good day, class. So for our meeting today, I'm going to discuss lesson 4, which would tackle about systemic mycosis. So among the different types of mycosis class, systemic mycosis is the one that is considered the most dangerous, the one that is also considered the most deadly, and it tends to have a high mortality rate. But it has a weakness class which later I will explain kung ano. Now, systemic mycosis class in this lesson is that we're going to discuss a variety of infections, each ranging in severity from subclinical to progressive debilitating disease. When you say debilitating disease, this is a type of disease that would paralyze the body. So from subclinical, when you say subclinical class, usually asymptomatic, wala kang makikitang symptoms sa kanya, to even progressive. So when you say progressive, from a single cell or from a single tissue, it can spread to the entire body. Now, some of the species that we are going to discuss here, class, are dimorphic, meaning they would grow in infectious mold form in the environment, but would switch to a round yeast-like form in infected human tissues. So sa environment class, they are in the mold form, but once they start infecting human tissues, they would switch or they would morph to their yeast form. Now, they differ from opportunistic fungi in their ability to cause disease in previously healthy person. In opportunistic fungi kasi class, they would only infect or they would infect immunocompromised immunocompromised individuals. But for systemic, even if you're healthy, there's a possibility that you could get infected. However, the most serious infections class would still occur in patients with compromised immune system. Each of these fungi class is restricted to geographic niches corresponding to the environmental habitats of the mold form of the species. So this is their one weakness class. They are considered endemic. They are restricted class to their geographic location. They are not easily spreadable to other regions. So some of them are only located in North America, in South America, something like that. So that's one weakness of this type of mycosis. They are restricted to a certain geographic location. Now, the systemic mycosis class that we're going to discuss, there are four. We have your Coccidioidomycosis, your histoplasmosis yan class, Blastomycosis, and Paracoccidioidomycosis. All of these class are systemic mycosis that are geographically restricted to specific areas of endemicity. So let's start with histoplasmosis. So histoplasmosis class is caused by the fungi Histoplasma capsulatum. Its habitat class are avian and bat to bat, BAT. Bat habitats, especially from their guano. When you say guano class, these are the feces of bats. So usually you would acquire histoplasmosis if you go to caves, mga kuweba. 'Di ba ang mga bats nyo, they would live on the roof or on the top of caves. Then they would defecate inside the feces of or the guano of the bats is where you can find your histoplasma capsulatum. So if you inhale the spores of the mold, there's a possibility that you could get infected. Now, the tissue form of the histoplasma class is oval. Oval, it is an oval yeast with a size of 2 by 4 micrometer. And it is known to infect or usually found inside macrophages. Found inside macrophages. Then we have your Coccidioidomycosis. This is caused class by the fungi Coccidioides posadaci and Coccidioides immitis. Their habitat class are dry soil. In the tissue form, they appear as spirules. 10 to 80 micrometer, and the spherules class would contain endospores that are 2 to 4 micrometer. Then we have your blastomycosis. Now, blastomycosis class is caused by or blastomyces dermatitidis. Habitat is river banks. Tissue form class is that they are a thick-walled yeast with a broad base, usually single and buds 18 to 15, 8 to 15 micrometer. Then we have your Paracoccidioidomycosis. This is caused by your Paracoccidioides brasiliensis. Its habitat is the soil. The tissue form is that they appear as large multiplying yeast 14 to 30 micrometer in size. Now let's start with the first one class, the histoplasmosis. So this is caused by your Histoplasma capsulatum. This is a dimorphic soil saprophyte that would cause histoplasmosis. This is the most prevalent pulmonary fungal infection in humans and animals class. So please take note of that. In nature, Histoplasma capsulatum grows as a mold in association with soil and avian habitats, being enriched by alkaline nitrogenous substrates in guano. Histoplasmosis class is also known as your Darling's disease. So let's say, 'di ba yung guano ng bats, they would go to the soil. Now, in the soil class, the molds of Histoplasma capsulatum would develop and grow. Now, accidentally, you disturb the soil. Nandun ka sa cave. You were walking, then you accidentally disturb the soil with guano, which contains histoplasma. After disturbing the soil, nagkaroon ng alikabok, there would be dust, and you would accidentally inhale, class, the spores. What happens is that inside the body, the spores would become yeast. They would become yeast, and what the body would do in response to the presence of yeast is that it would activate its our immune system. And one of the first responders of your immune system class as a part of your active immunity is the cellular components. And a part of the cellular components class is your WBC, such as your macrophage. And if you're aware, ang macrophage nyo is capable of performing phagocytosis or cell eating. So ang gagawin ng macrophage nyo is that the macrophage class would phagocytize, excuse me, would phagocytize the yeast form of Histoplasma capsulatum. That is why when you try to view samples of tissues or samples positive for histoplasma class, you can find macrophages that would contain histoplasma capsulatum. So that's the relationship between macrophage and histoplasma. Now, at temperature below 37 degrees, primary isolates of Histoplasma capsulatum class would often develop brown mold colonies, but the appearance would vary. In tissue or in vitro on rich media at 37 degrees, the hyphae and conidia class would convert to small oval yeast cells. So, ganyan yung appearance niya class. Small oval yeast cells in a in vitro, or when you say in vitro, you put it in a media. So there's another description class. At 25 degrees centigrade, they would appear as septate hyphae with round pyriform, PYRIFOR, pyriform microconidia on short branches or directly on hyphal stalk on prolonged incubation. On prolonged incubation class, knobby tuberculate macroconidia would form. So take note of this. So 25 degree or in their mold form class, they would have a septated hyphae. So tingnan niyo yung hyphae dito. They would appear septated with round pyriform microconidia on short branches. Now, if there is a prolonged incubation class, sobrang tagal mo siyang in-incubate sa room temp, siguro mga 1 month, there would be the development of this snobby tuberculate macroconidia. Malalaki. Then at 37 degrees class, they appear as small budding yeast within mononuclear or macrophages. So this is an example of a monocyte or a macrophage class. This is a macrophage. And inside the macrophage class, you would find yeast cells. So ito yung yeast cells na yan. This is your histoplasma capsulatum engulfed by the macrophage. Now, what are the lab diagnosis class? So, for specimens use our sputum, urine, scrapings from superficial lesion, bone marrow aspirate, and you could even use the buffy coat of your whole blood. For microscopic exam, you're going to use fungal wall stains to visualize small ovoid cells. You could also use Gomori methenamine silver or your periodic acid shift and even GEMSA. So you could use GEMSA class for the bone marrow aspirate saka Buffycoat. Then for the culture class, you would use glucose cysteine blood agar at 37 degrees. So for the yeast form. Then for the mold form, you could use SDA or subrhodextrose agar or IMA or your inhibitory mold agar. So do take note of that. Now, what are the treatment, prevention, and control for histoplasmosis? So acute pulmonary histoplasmosis class is managed with supportive therapy and RIS. The use of itraconazole is the treatment for mild and moderate infection. Now, in cases there is a disseminated histoplasmosis, systemic treatment with amphotericin B class is often curative, though patients may need prolonged treatment and monitoring for relapses. Patients with AIDS would typically relapse despite therapy that would be curative in other patients. Therefore, AIDS patients require maintenance therapy with itraconazole. So, dalawang antifungal lang class ang ginagamit dito. It's either itraconazole or amphotericin B, which both of them are very strong antifungals class. Now let's go to the second one, your coccidioidomycosis. Now, coccidioidomycosis is caused by your Coccidioides posadacea and imitis. They are phenotypically indistinguishable. They would cause similar clinical manifestation and are not differentiated in the laboratory. So they are phenotypically indistinguishable class, meaning same sila ng appearance. What you need to do is that you would need to perform a genotypic test or you would test for the genotype. So you're going to do a molecular test, yung PCR ganon, for you to be differentiate Posadacea from imitis. Now, acute primary infection with C. imitis is often asymptomatic. Take note of that. Ha? Acute, when you say acute class, this is a new infection. They are often asymptomatic. But it can manifest class as a complex of symptom called valley fever by the residents of endemic area. Symptoms of valley fever class would include fever, malay, dry cough, joint pains, and sometimes a rash. Ito yung rash na class. So take note of that. This is known to cause the valley fever or yung coccidioidomycosis. Now, what is the morphology of C. posadacea and imitis class? So, these agents would produce a white to tan cottony colony. The hyphae would form chains of arthroconidia or arthrospores, which would often develop in alternate cells of hypha. These chains fragment into individual arthroconidia, which are readily airborne and highly resistant to adverse environmental conditions. The small arthrochonidia would remain viable for years and are highly infectious. So later class, may picture dyan, I will explain the life cycle. Following their inhalation, the arthrochonidia would become spherical and enlarge, forming spherules that would contain endospore. Now, the problem with your coccidioidomycosis class, this is a major biohazard to laboratory personnel. As much as possible, class, when you handle coccidioidomycosis, you need to work on a biosafety level 3 guidelines and protocol. So at room temp, they appear as stick-walled, barrel-shaped arthrochonidia that would alternate with empty disjunctor cells. At 37 degrees, they appear as stick-walled spirules filled with endospores. So para mas maintindihan nyo class, so for a better understanding on the life cycle of your coccidioidomycosis, ito yung diagram niya. Now, coccidioidomycosis mold form class would typically reside on soil. Now let's say you're walking in a trail, nagja-jogging ka or nagha-hiking ka. You accidentally disturb the soil class. You accidentally disturb the soil. Upon disturbing the soil, arthrochonidia, the arthrochonidia of your coccidioidomycosis would be inhaled. Now, after inhalation, class, the arthrochonidia inside our body after 24 hours would enlarge. When you say enlarge, it would grow in size. After 48 hours, class, it starts to divide on its own. It starts to divide on its own until after 72 hours, it becomes a spirule. So from an arthrochonidia, from a barrel-shaped arthrochonidia, it would become a spirule on day 3. Now, once the spirule matures, pag nag-mature siya, class, if it becomes mature, it would start to form endospores inside the spirule. So, these circles found inside the endospore are the spirules. Now, after 5 days, class, two things can happen. The spirules would be released. So, pag umubo yung patient, if the person would cough, the patient would cough, the endospores would be released. Or what happens is that the endospores will continue to stay in our body and it would continue to multiply. Padami siya ng padami until the patient becomes a chronic sufferer of coccidioidomycosis. Now let's continue the cycle. Once the patient coughs out spirules containing endospores or the endospores class would start to develop tubular structures. Nagkakaroon siya ng tubular structures. Now, these tubular structures are part of the mold form. Para siyang hyphae. The endospores now would continue to become a mycelium. Till it's become a mycelia, and the mycelia class would start to form the arthrochonidia. So, ayan yung arthrochonidia nyo. Arthrochonidia would form as alternate cells along the mycelia. Then it starts to disintegrate, releasing arthrochonidia, where it would remain in the soil for a very long time until the cycle would repeat itself. So it's very dangerous class itong mga systemic mycosis na to because you could acquire them via inhalation. That's the problem with systemic mycosis. Their mode of transmission is inhalation. If you do not wear the proper PPE, you could get a type of pulmonary disorder that is very difficult to treat. Now, what are the lab diagnosis class? So specimens would include sputum, exudate from cutaneous lesions, spinal fluid, blood, urine, and tissue biopsies. For the microscopic exam, you could use KOH, fluorescent stain calcofluor white, histologic stains, hematoxylin and eosin, Gomori methenamine silver, periodic acid chip stain. For the culture, you could use inhibitory mold agar, BHI blood agar slant. This is slant class. Incubate at room temp or 37 degrees. So pag IMA, this is for the mold. For BHI blood agar slant, this is for the yeast. So take note of that. Now, what are the treatment and prevention and control for coccidioidomycosis? In most persons, symptomatic primary infection is self-limited and would require only supportive treatment. Although itraconazole may reduce the symptom, however, patients who have severe disease would require treatment with amphotericin B, which is administered intravenously. So if you're having a very weak infection class with coccidioidomycosis, itraconazole would be enough. But if it's a severe type, chronic na siya, you need to use amphotericin. Cases of coccidioidal meningitis have been treated with oral fluconazole, which has good penetration of the central nervous system. However, long-term therapy is required and relapses have occurred. So that's the problem, class. There are cases wherein relapses, uulit yung sakit. That disease is not communicable from person to person, and there is no evidence that infected rodents would contribute to its spread. Some measures of control can be achieved by reducing dust, paving roads and airfields, planting grass or crops, and using oil sprays. Now let's go to the third type of systemic mycosis class, the blastomycosis. Now, the blastomycosis class is also known as your North American blastomycosis or Gilchrist disease. It is caused by the fungi Blastomyces dermatitidis. It is a chronic infection with granulomatous and suppurative lesion that is initiated in the lungs. Hence, dissemination may occur to any organ, but preferentially to the skin and bones. The disease has been called North American blastomycosis because it is endemic, and most cases class would occur in the United States and Canada. The blastomyces this dermatitis class, please take note, is a very thermally dimorphic fungus that would grow as a mold in culture, producing hyaline and branching septate hyphae and conidia. At 37 degrees or in the host, it converts to a large, singly budding yeast cell. So when you grow your blastomyces class on SDA at room temp, a white or brownish colony develops with branching hyphae bearing spherical, ovoid, or pyriform conidia. But if you grow it on tissue or at culture at 37 degrees centigrade, please take note, class, that they would grow as thick-walled, multinucleated, spherical yeast cell that would usually produce single buds. So to simplify, class, sa room temp or the mycelial phase, they would form a septate hyphae with round or pyriform conidia that would resemble lollipops. So take a look at the picture, class. So this is their mold form. They, yung conidia niya, they would resemble lollipops. Then for 37 degrees or the yeast phase, they would appear as a thick-walled large yeast cell with a single bud on a broad base. So makapal yung base niya, class, tapos magpuproduce lang siya ng single bud. So parang ganito. Yan. Then this is the single bud class. Now, what are the lab diagnosis class? So, specimen would include sputum, pus, exudates, urine, biopsies from lesion. For microscopic exam, wet mount specimens may show broadly attached buds on thick-walled yeast cells. This is apparent in histologic sections. Culture, usually colonies would develop within 2 weeks on STA or enriched blood agar. at 30 degrees centigrade. Treatment, prevention, and control class. Severe cases of blastomycosis are treated with amphotericin B. In patients with confined lesions, a 6-month course of itraconazole is very effective. Blastomycosis is relatively common infection of dogs class in endemic areas. They cannot be transmitted by animals or humans. Unlike Coccidioidomycosis and Histoplasma capsulatum, Blastomyces dermatitis has only rarely been isolated from the environment. So very rare mo siyang ma-isolate sa environment class. That's why its natural habitat is unknown. However, the occurrence of several small outbreaks has linked Blastomyces dermatitis to rural riverbanks. Do take note of that. Let's go to the last one, your Paracoccidioidomycosis. This is a progressive mycosis class of the lungs, the skin, the mucous membrane, the lymph nodes, and internal organs. This is caused by the fungi Paracoccidioides brasiliensis. Now, Paracoccidioides brasiliensis class is the thermally dimorphic fungal agent of Paracoccidioidomycosis, or also known as the South American Blastomycosis. That's why kaya siya tinawag na South American Blastomycosis class. It's endemic to Central and South America. P. brasiliensis is inhaled and initial lesions would occur in the lungs. So after inhalation sa lungs class, pagkakaroon ka ng mga fibrosis or lesions sa lungs. After a period of dormancy yan, that may last for decades, the pulmonary granulomas may become active, leading to chronic progressive pulmonary disease or dissemination. Now, morphology class. Cultures of the mold form of pre-Brasiliensis would grow very slowly and would produce chlamydospores class. So this is the one that is known to produce your chlamydoconidia or chlamydospores. The features are not distinctive. At 36 degrees on rich media, it forms large multiple budding yeast cells. The yeasts are larger and have thinner walls than those of Blastomyces dermatitis. The buds are attached by a narrow connection. To simplify class, ito lang yung tatandaan nyo dyan. At room temp, they have terminal chlamydoconidia. On yeast form, they appear as yeast with multiple buds resembling a ship's wheel or a mariner's wheel. So pag tinignan mo yung yeast form niya, if you look at the yeast form, it looks or it would resemble a ship's wheel or a mariner's wheel. Diba yung sa barko class? Diba may ganun-ganun yan. So yun yung appearance niya. Laboratory diagnosis class. So microscopic keys are often apparent on direct microscopic exam with KOH or calcofluor. Culture class, culture of SDA or yeast extract agar are incubated at room temp and confirmed by conversion to the yeast form by in vitro growth at 36 degrees centigrade. Treatment, prevention, and control class would include itraconazole, which would be the most effective, but ketoconazole and trimethoprim sulfamethoxazole are also efficacious. So effective din sila class. Severe diseases can again be treated with amphotericin B. Since P. brasiliensis has only rarely been isolated from nature, its natural habitat has not been definitively determined. As with other endemic mycosis, Paracoccidioidomycosis is not, communicable. So here are the references for this lecture. Thank you.